Okay, time for the last chunk here. So if we remember, the whole point of doing cellular respiration is to actually harvest energy from glucose and then use that energy to create ATP. Making ATP is the whole point here, and we haven't made very much at all yet. So there's the question: how does the energy from the high energy electrons that we have harvested so far from glucose get converted into the chemical energy in ATP? So just to remind you, we have all these high energy electrons, here they are, in all these taxis called NADH that we have harvested and created through the initial 3 steps: glycolysis, transition, and the citric acid cycle. Next, we have to make the ATP. So let's go through and see what happens. All right, so this looks a little complicated, so let's go through this slowly. What we're looking at here is the inner mitochondrial membrane. This phospholipid bilayer is the inner mitochondrial membrane, which is the one that's all folded up. This pale yellow area is the matrix, so the very inside part of the mitochondria. And this dark yellow area is the intermembrane space, which means the outer membrane of the mitochondria is out here. You can't see it. So it's over here. All right, here's my NADH. So that's my taxi full of electrons. All right, it's coming up to this first protein here. This is a chain of proteins, these blue blobs, and they're embedded in the inner membrane of the mitochondria. So here's the matrix. My NADH, my taxi, which is carrying the electrons from glucose, is coming up here, and it's going to dump out its passengers. When it empties itself, it turns into NAD, and it can go off like all taxis do to find new passengers. All right, so here's my high energy electrons, and they're going to be given to this first protein in the chain. So this protein can accept these electrons. This protein then passes the electrons to this molecule, which then passes them to this molecule, which then passes them to this molecule, which then passes into this molecule. So it's like a bucket chain, and these electrons get passed down from molecule to molecule to molecule. As they pass from molecule to molecule, this yellow line is the path of the electrons. These proteins use some of the energy from these electrons, get sort of sucked into these proteins and used to power a pump, to pump hydrogen ions from the matrix into the intermembrane space. So remember, these hydrogen ions, they're charged. They can't just cross through the membrane on their own because they're charged. And they're getting forcibly pushed by a pump into this space. They have to be pumped because there's already a lot of them here from all these other pumps that are pumping hydrogen ions. So what we're trying to do here is accumulate a high collection or concentration of hydrogen ions in the intermembrane space. We're piling up hydrogens in this space, trapping them in this little space here. As these electrons are passed, gradually as they move from protein to protein to protein, more and more and more of their energy is squeezed out of them. This is those juicy grapes I was telling you about. We're squeezing all the juice out now. The juice is the energy, and the juice or the energy is being used to push hydrogens into the intermembrane space. Push the hydrogens, push the hydrogens, push the hydrogens. By the time we get to the end of the ETS here, the last protein in the chain, these electrons that were high energy, they're nice and red and full of high energy, by the time they get to the end, they've been squeezed dry of all their energy. There's still electrons at the end, but now they are low energy electrons. They're just black and boring electrons with no energy in them. They're still negatively charged particles, but they're not super vibrating and super energized. And so, and this is like the skins of the grapes once all the juice has been sucked out of them or pressed out of them. So what we have to do is get rid of these electrons. We don't need them anymore. We've used their energy. And so we have a molecule that we know loves electrons from way back in Unit 1. And here it is, oxygen. So oxygen is going to swoop in here. Oxygen doesn't care if the electrons are high energy or low energy. It just wants electrons, period. So it's going to come and grab these electrons coming out the end of the electron transport chain. it's like the garbage collector, or taking away the skins of the grapes. And the oxygen picks up the electrons, it picks up a couple hydrogen ions too, and does a chemical reaction and turns into water. So if you remember that overall equation for cellular respiration, it needed 6 oxygens and it created water as a waste. And that's where this happens. Here's the oxygens, they come in, they pick up the electrons at the very end of the electron transport chain, they're taking away the waste electrons in the form of water. All right, and then this water can kind of go off and join the rest of the water in your body, or you can pee it out or breathe it out. It's just water at this point. It's the end product of this whole thing. All right, so we have taken apart glucose, harvested its electrons, ran its electrons through this electron transport chain where we sucked out all their energy, and then we got rid of them by giving them to oxygen, and that turned it into water, and off they went. And we were left with all these hydrogen ions that we forced up into the intermembrane space as part of this process. So let's see, get this to go next. Here go my electrons. Off they go with water, and the last stage is right over here on the far right side. So this molecule should look familiar to us from the previous stuff we learned about photosynthesis. This is an enzyme. This is the molecule called ATP synthase. It's an enzyme. Its name ends in A's. It synthesizes ATP. And what's the whole point of cellular respiration? To make ATP. And ATP synthase is the best, most efficient way that nature has come up with making ATP. However, to make ATP synthase work, it has to have hydrogen ions going through the middle of it. Remember that? It spins around as hydrogen ions go through it. So to make ATP synthase work, you have to have a hydrogen ion gradient where the hydrogen ions want to move from one place to another. And that's exactly what we created right here. If you remember, we spent all this time pushing hydrogens into this intermembrane space where they're trapped because they can't get through the membrane on their own. And then the mitochondria very thoughtfully provides an exit route for them. Once again, this analogy of the football stadium. Everybody's crammed in the stadium. At the end of the game, there's only a couple of ways out, a few turnstiles to go through, and everybody streams through the turnstiles, and as they go out and diffuse off to the back to their homes, they spin the turnstile as they leave the stadium. And that's what's happening here. The hydrogens want to escape where they're all crowded in here. They're going to move through the ATP synthase. As they move through the ATP synthase, this direction, they're going to make it spin around. And as it spins, it's going to grab ADP and a P and stick them together to make ATP. And that's the whole point of this. And this is incredibly efficient, this part. So we do steps 1, 2, 3, glycolysis, transition, citric acid cycle. We don't make much ATP, but what we are doing is getting all the fuel to run these pumps. Those electrons are the fuel. And then the very last step, we run all these pumps, create this gradient, harvest the energy from the gradient with this enzyme, and bam, there's all our ATP at the very, very last part. And then we get rid of all the trash, all the spent electrons are taken away by oxygen in the form of H2O. That is that. That's the end of cellular respiration, the 4 main steps. And I'm going to end it there.